Okay, today we're going to talk about human endogenous retroviruses, but to appreciate what those are, we need to know a little bit about actual real retroviruses. So I'm going to talk about HIV as an example first. Okay, so here's HIV, a real retrovirus. It is represented in its DNA form as it exists in the chromosome when it would be sitting in an infected cell getting ready to produce new virions. Okay, now this diagram is very representative of basically all retroviruses, of this type at least, in the way the genome is organized. On the very left, we have the LTR, long terminal repeat, and there's another copy of that exact same sequence on the right. So that's the characteristic of this type of transposon for sure. So they all have that, and they all also have a genomic region that's divided up into basically three parts called GAG, Pol, and ENV. This is a very characteristic genome organization. This is HIV, and it does have additional regulatory proteins, VIV, VPR, etc., in addition to those standard general retroviral genome organization. But this is very typical of the genome. Okay, so the GAG, Pol, ENV organization, flanked by the long terminal repeats, is very typical. But the virion, of course, is going to contain RNA, not DNA, and it will get reverse transcribed in the process of infecting new cells. Okay, so there are a lot of retroviruses that are very much like this, including, just for example, mouse mammary tumor virus, avian leukosis virus, human T lymphotrophic virus, etc. So these are infectious. They produce infectious particles that are transmitted from cell to cell and from individual to individual, as viruses do. Okay. Another general feature is that the LTR, left LTR, contains the promoter. So this is the only promoter that's present in these viruses, and so they use this one promoter to produce the RNAs that will make not only the new copies of the viral genome for incorporation in infectious particles, but for their own RNAs to produce protein. So we're going to have the left LTR as an important feature of these viruses. So there is a complicated splicing pattern that goes on here, all beginning with the LTR. Okay, so what happens is the full-length virus particle will end up, some of it at least, being part of the new virions that are going to be produced by an infected cell. So that will be a full-length RNA, which can be used for virions, but some of it is also used for translation of proteins. So the splicing patterns are again characteristic of this class of viruses, where you have your full-length one, but there's also a splice which leads to the production of the ENV region of the viral genome in the form of an mRNA that can be translated to produce the two proteins from the ENV region, which are the transmembrane protein and the surface protein. So this type of splicing pattern is very characteristic of these. Now we do have separate splicings for the regulatory molecules, which is, in this case, distinct for HIV. Okay, so retroviral protein production is really weird. The weird fact is that the retroviral proteins are translated initially as what are called polyproteins. So when we talk about the GAG, Pol, and ENV, those are the initial proteins that are translated, but they have to be proteolytically digested, processed, to form the final proteins that are going to be actually part of the virion. Okay, so the protease that's responsible for this is encoded by the retrovirus as part of the Pol region of the genome. Okay, so the GAG region is for capsid, matrix, membrane glycoproteins are from ENV. Okay, so what this looks like is, if we consider the GAG region that's present in the longer of the full-length RNA, that will be translated to form a protein, which is the precursor to the eventual core structural proteins, the P18, 24, and 15. So that is one translation product which counts as a polyprotein, because it will be ultimately producing three different proteins from that single initial protein. Same for the Pol region, that is going to produce a precursor, P160, which will then be proteolytically cleaved to form the protease itself, but also the reverse transcriptase and the integrase, which are, of course, crucial parts of the viral life cycle to get it to go and insert in a new location in the genome. or some other genome. Finally, the M region produces by a precursor, which is the M precursor, which is then proteolytically cleaved by the protease to form the GP120 and GP40, which will end up being on the surface of the virus extravirally and are used for infectious purposes. Okay, so the GAG region becomes the capsid and nucleocapsid. Pol forms reverse transcriptase, integrase, and protease. N will form the surface proteins of the virion, and then the regulatory proteins are actually translated separately. So one important feature of these is that these proteases are very important for the virus, obviously, and that's why protease inhibitors have become really crucial anti-HIV drugs, because the virus needs to have this protease to produce its active reverse transcriptase, for example, in order to continue the life cycle. So these are very effective inhibitors. Here's our splicing patterns again. Okay, and that shows where the proteolytic cleavage sites are. In the GAG, the little cleavage sites are shown, pro, pol, cleavage sites, etc. So those initial protein products are processed proteolytically to produce the final one. And that's very characteristic of this type of virus. Okay, so what these are retroviruses which no longer go from cell to cell or from person to person or organism to organism, but rather viruses that have taken up residency in our genome and remain there. They don't transfer at all. They're mostly decayed in the sense of having a lot of mutations in them, most of them have little protein coding capacity, but some can be transcribed. Okay, most of them are not active at all, and the only reason you can tell that they're there is just looking at their sequence, comparing them to existing retroviruses. But they're not all completely dead. They can have functions in cells that we'll see. Okay, so in terms of classification, they are classified according to their ages and in what organisms particular ones are found. The oldest ones are the HERV-L group, which apparently colonized but the mammalian genome very many years ago. Then we have the HERV-K class, which came later, the HERV-W class, and finally there's some more recent HERV-K insertions, which occurred approximately 670,000 years ago. So these have been around for a long time and are characteristic of mammalian genomes in general, but we have maybe more than a lot. Okay, so they're now a permanent part of our genome. They inserted somewhere in the distant past, and they are not active as viruses, but they're still there. They are inactive in the sense that there's no completely active element where all of the open reading frames are still intact. But there do exist examples of, for example, the reverse transcriptase and the N for sure, that are essentially intact. Within the genome, where we have 1,000 copies, there is at least one essentially active version of all of the viral proteins. Okay, there are some examples of complete ORFs, but no intact actual virus. Okay, so these are vertically transmitted, which is to say they're passed from parent to offspring, and they're not infectious. Okay, so here's the vertical transmission idea, comparing what happens to a normal virus versus what happens with these guys. Okay, so in a normal virus, you have a virus, and somebody gets infected with it in their somatic cells. There, the virus can replicate from one cell to another, passing itself within the body, but it can also be transmitted to another person. So this is the normal way in which viruses go about conducting their lifestyles. In contrast to this, our HERVs are transmitted vertically, which is to say from parent to child. That at some point in the distant past, they were inserted in the germline of the person, our predecessor, and have remained there. So they're there in our genome, and they're passed down like any other Mendelian type DNA sequence. So one doesn't get these from the outside or from anybody else. They're passed to us from our parents. So this is to convince you these are real retroviruses. Here's an example, a different sort of diagram, but it shows that it has all the expected features of a retrovirus. It has a 5' LTR, or left, has a 3' LTR. It has a GAG, POL, M regions, just like HIV does. The protease is a little bit different in its translated from a slightly different way, but it's still right there in its customary location among retroviruses. So this certainly looks like a retrovirus, no question about that. Okay, it also has a splicing pattern, which is what you would expect from a retrovirus. You can have full-length RNAs produced from a promoter in the 5' LTR, encoding GAG, pol, and ENV, not ENV, GAG, pro, and pol. And then there are the spliced versions whose job it is to produce ENV proteins. So this looks, for all the world, like a retrovirus, easily recognized as being similar to things like HIV. Okay, so in terms of its interactions with our genome, there's so much of it, it must be doing something. Well, a lot of the copies are actually just LTRs. Like, they don't have the GAG-POL N part, they just have the LTRs. So these are called solo LTRs. And so they have no protein coding capacity. On the other hand, they do contain the promoter, which would have driven the retroviral RNA expression. And so they can actually become activated some of the time. Now, mostly they're silenced with the usual sorts of, histone modifications that are silencing and 5-methylcytosine, etc. But they can actually become activated under some conditions. And they can, under some circumstances at least, influence the transcription of nearby genes, which can be of concern. Okay, so here's a particularly weird example of interaction between this retrovirus and our own genome, and that's the existence of these proteins called syncytins. Well, it turns out that since the ENV proteins are the source of these syncytin proteins that are actually used during formation of the placenta during early embryonic development. Okay, this is bizarre. Okay, so the retroviral ENV protein is what is used by the virus to bind to and fuse itself with a new host cell membrane. So these are considered fusogenic because they facilitate membrane fusion during the viral infection of a new host cell. So in retroviral infections, the ENV proteins, the surface and transmembrane proteins, do that exact function. They facilitate the fusion between the virion membrane and the cell surface. Well, it turns out that we have actually co-opted or X-adapted these proteins for use during our own embryonic development because these proteins are actually produced during early embryonic development, during which time they actually facilitate the fusion of cells to form the placenta. Now, this is totally bizarre. Okay, so looking at what these proteins look like and thinking about what they do, these are derived from the retrovirus. We've got the syncytin 1 has the surface part, the transmembrane part, the SU and the TM from the ENV region, and there's actually a proteolytic cleavage that occurs just as if it were within a retroviral particle itself. So these are retroviral proteins, really. So the connection here is that if you consider what the retrovirus is going to try to do when it's going to try to infect a new cell, it has its surface protein, which recognizes a receptor on the susceptible cell, binds to it, and that promotes fusion between the external part of the virus, which is a membrane, and the membrane of the new host cell. So this is a natural part of retroviral infection, whereby the capsid is then delivered to the host cell for the purpose of starting another infectious cycle. Well, that same thing happens during cell fusion, where you have the ENV protein sitting on the surface of a cell, recognizes a receptor on another cell, and in this case, the membrane fusion event leads to the fusion of the two cells, where you form a syncytium, which is, say, a cell that has more than one nucleus in it. And this type of cell fusion event occurs during formation of the placenta and is actually guided, at least in part, by these ENV proteins from our endogenous retroviruses, which is completely, utterly bizarre. So this membrane fusion thing has been used, and this is not the same virus, but other mammals have the same process. Now, this process of forming syncytia, which is to say cells that have been fused with each other, can be a problem, particularly if you want to create cancer cells, etc. If you've got these being expressed on the cell surfaces, you could have syncytium formation under conditions where you don't want actually to do that. So this is maybe one of the more bizarre experiments I've ever heard of in my life. And the idea is that it looks like a retrovirus, but it's not really because there aren't any active ones. So these people decided they would try to create an active one by comparing the sequences of the known ones that we have in our genome. We know they've got mutations in them, but if you can compare them, you can kind of get a consensus of what the ancestral or unmutated sequences must have been. So by comparing all the different, or maybe a large number of different forms of this retrovirus in our genome, they were able to derive a sequence of what we think an active one would look like. So they created that, basically getting one element, mutagenizing it to fix whatever mutations are there, and to try to decide if this thing was infectious. And in fact, they did do that, and it is actually infectious. Why they did that, I don't know, but they did. Anyway, the resurrected virus apparently forms virus-like particles in the cytosol that you can see budding off the surfaces of infected cells, and it can actually be used to infect new cells. Okay, so what this is representing here, all those Ks aren't lysines. Those are examples of the various isolates that were used to construct this consensus of what the actual one must look like. But they did get a consensus based on mutations by comparing the sequences of all these different ones. So this is what it looks like if you make this and put it in cells and look in the EM, you get virus-like particles forming inside the cells and even budding off the cell surface. So it looks for all the world like a real retrovirus, which it is. And so just to be even more convincing, they isolated some of these viral particles from supernatants of infected cells, and they were tagged so they could see who was where, and then use those to infect new cells to prove that they're actually infectious. Well, they got some infections, and then characterized the locations where the new virus had inserted itself. And one, of course, characteristic of this type of transposition is that you get target site duplications. And so they characterized the sequences immediately adjacent to the inserted element on both ends and saw that whatever sequence was there was in fact duplicated, much as if it were a real infection, because it is a real infection. And by comparison to naturally occurring ones, which also have target site duplications. So yes, they woke it up. And so it's a real retrovirus, or can be made to be a retrovirus. Okay, so it did insert itself into genomes of recipient cells, and they have the target site duplications. So this is presumed to be very similar to whatever the actual ancestral retrovirus was that originally colonized our genome. So we can learn more about what that retrovirus looks like that no longer exists by examining features of this particular newly awakened retrovirus. Too weird. Okay, so in terms of what's going on with these in our cells, there's really a lot of controversy about this. Like, they are transcriptionally active some of the time, or at least some of them are, but there are 1,000 of them. Okay, they're normally silenced and don't produce RNA, but sometimes they do. And often the encoded proteins are defective because they've got mutations. And also the fact that a lot of them don't have any actual protein coding sequence at all, they're just solo LTRs. On the other hand, there's some concern that they actually could contribute to the formation of certain diseases, but the evidence is it's not out there yet. Okay, so there's a lot of circumstantial things that go on with these. It is known that the ENV transcripts are upregulated in certain cancers, especially breast cancer and in some forms of prostate cancer. In general, people have not been able to establish a causal effect of this kind of overexpression in any particular cancer. It could be an effect of the cancer, or it could contribute to the cause of the cancer. So it's really very much not known about this. People want to make statements about this might be true or this could happen, but there's not a lot of there's certainly no clear case in which a causal relationship has ever been determined. But it's pretty interesting stuff anyway. Okay, so now we're going to have some sample questions. Oh yeah, that's the end of our test material. Yay. And so these are just some sample questions from old tests. Okay. And I don't think I'll go through these right now because they're here. I've got a slide with the answers. Slide, 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 answer, answer, answer. And so I'm going to leave these in the PowerPoint for the purpose of review. But these are also going to be there posted elsewhere. Right, so we've got dicer. We've got 2-O-methyl ribose. This is what the cap structure looks like, remember? And this process is, of course, alternative splicing, and the sex-lethal protein causes exon skipping. Yay. Okay.